start with restriction endonucleases. We talked about restriction endonucleases before, and we said that nucleases are enzymes that cleave nucleic acids, DNA and RNA. Specifically, you have endonucleases, which are enzymes that cut DNA in the middle of the molecule. Okay, this is versus exonucleases, EXO nucleases, which degrade, cleave the nucleotides of DNA from either end. So you have these endonucleases. Specifically, we're going to talk about restriction endonucleases, which are enzymes, bacterial enzymes, that degrade DNA in the middle at specific positions. Okay, so what they do is that they recognize a specific sequence called a restriction site, and usually it is about 4 to 8 nucleotides long, or base pair long, and they cut within these sites, resulting in the formation of restriction fragments. Okay, so right here you have a DNA, you have a restriction nuclease, it recognizes a restriction site, CCC, GGG specifically, and this enzyme would then cut the DNA within the restriction site, resulting in the formation of restriction fragments. So what they do is that they cut phosphodiester bonds. Okay, notice something, that these sequences can be read from 5' to 3' on both strands, the same exact thing. So EcoR1, that's one type of a restriction endonuclease, very famous. We talked about it before, and I said that EcoR1 recognizes this sequence, G, A, T, T, C. Now, if you look at the opposite complementary strand, notice that the sequence is exactly the same, G, A, T, T, C. Again, we're reading from 5' to 3'. Look at another endonuclease, Hindi3. It's called Hindi3. AAGCTT. The opposite complementary strand, AAGCTT. Same exact thing. Here's another one, SMA1, CCCGGG. The same thing is read from 5' to 3' on the complementary strand. Now, these sequences are known as palindromic sequences. Palindromic sequences, that is, sequences that are read from either on either strand, the same exact thing. Okay, so what happens here is that these endonucleases would make two types of cuts depending on where the cuts are. One cut is known as a blunt cut, resulting in the formation of blunt-ended fragments. The meaning of blunt is that it's sharp. Okay, it's a cut that goes straight from up to bottom. Okay, so for example, you can have a cut that goes like this, like a knife. Okay, so straight cut, resulting in blunt-ended fragments. That is, there are the ends are smooth. This is in contrast to what is known as staggered cuts. What staggered cuts mean is that you have a cut like between G and A, right here on that strand, and on the other strand, the cut occurs between G and A, resulting in the formation of what is known as sticky ends or cohesive ends. Okay, the reason is that the reason why they're called this sticky ends is that this end is complementary to this end. So these two ends can form hydrogen bonds between each other, except that it is not stable. Why? Because there is no phosphodiester bond between this nucleotide and that nucleotide. Okay, so that's the meaning of sticky-ended DNA fragments. Well, here's another look at it. A cut between G and A, resulting in the formation of two restriction fragments with sticky ends. So eventually this end can form hydrogen bonds with this end, except again that there is no phosphodiester bond. Okay. Now we can form a phosphodiester bond between the G and the A by an enzyme known as a DNA ligase. Now, the action here is known as ligation. It's like gluing the two strands with each other or the two fragments with each other. Okay? And the enzyme is known as a DNA ligase. Okay, so that's what a DNA ligase does. It comes in, it forms a phosphodiester bond between the 5' end of this fragment with the 3' end of that fragment. Notice the cohesive ends right here. Okay, so you can have the formation of hydrogen bonds followed by a phosphodiester bond formation between the two fragments. Now you have a DNA fragment that is linked, one continuous DNA fragment, and the ends are stable. Okay, remember that this enzyme requires ATP. It needs energy. Okay, so that's restriction endonucleases. Now, in the previous course, we talked about restriction endonucleases and how we can use these enzymes in RFLP, or restriction fragment length polymorphism, how we can differentiate samples from each other, or we can link samples to individuals because each individual has his or her own molecular profile. Okay, because of these differences in DNA sequences. So we talked about RFLP before and how we can determine paternity, or we can use it in forensic medicine, crime scene investigation, and so on. Remember that? Okay, so another use of restriction endonucleases is what is known as cloning. Okay, what cloning means is that you can make several copies of one thing. So if I say I want to clone a DNA, it means that I want to make multiple copies of this DNA fragment. If I say human cloning, it means that I'm making many copies of an individual. So that's what cloning means. Okay? Now, the thing is, cloning is really something natural in bacteria, because you can have a bacterial cell that is grown on a plate. You will study this in microbiology. It's called a petri dish. You can have this one single bacterial cell that eventually, after a few hours, or let's say 24 hours, it will form a colony. Now, each colony is really a clone of each particular cell. Okay, so these cells are all identical because they have the same DNA sequence or the same molecular profile. Okay, so this is what DNA cloning or bacterial cloning means. So DNA cloning is making copies of the same DNA. Well, how can we do that with DNA? How can we do DNA cloning? Well, the thing is, in order to do that, we need what is known as a vector. A vector is a carrier. Okay, so the idea here is that it's a carrier of the DNA fragment that we want to clone, that we want to make copies of. Okay, now what we do is that we use a vector, a carrier, and this carrier is usually, or more commonly, it's a DNA plasmid. A plasmid is basically an extra chromosomal DNA. What it means is that it's a DNA that is different than the bacterial chromosome. Remember, bacterial cells have one chromosome, one circular chromosome, but they can also have multiple copies of plasmids, which are extra chromosomal, okay, DNA pieces. That is, they can replicate independent of the chromosome, of the bacterial chromosome or the chromosomal genome itself. Okay, so a bacterial cell may have multiple plasmids. Okay, so a plasmid is a circular DNA. It's a small DNA that contains some genes, and these genes give advantage to these bacterial cells. Okay. Now, we take a plasmid and we insert the DNA fragment inside the plasmid. Now, so we have a DNA molecule having two different independent pieces of DNA integrated or ligated to each other. This is known as a recombinant DNA molecule. So a recombinant DNA molecule is basically a DNA molecule that is made of two different DNA fragments linked or ligated to each other. Now, the reason why I do this is that when I insert a DNA fragment like this one here, the plasmid is inserted into bacteria, and bacterial cells would make multiple copies of the same plasmid. So basically, we let bacteria clone the DNA, the recombinant DNA molecule. So instead of having, so if I insert it into a bacterial cell and this bacterial cell would make millions of other bacterial cells, I'm making not only millions but multiple millions of these of the plasmids, because each bacterial cell would make multiple copies of the same plasmid. Okay, so hopefully this will be clarified later on. So, so we use plasmids as vectors, as carriers. Okay, so bacterial cell would have one chromosome, but they can have multiple plasmids. Well, how can we make a recombinant DNA molecule? Well, we have to choose the right plasmid, and the right plasmid would have must have multiple features. First of all, they have to replicate, and they have to replicate independently of the bacterial chromosome, so they can make multiple copies of themselves. That's one. Number two, so in order to replicate, they have to have an origin of replication. Remember this? This is the site, a sequence that allows a plasmid or a chromosome to chromosome replication to be initiated. So the starting point is at the origin of replication. Another feature of the plasmid is that it must have a place whereby I can insert the foreign DNA. It must have a place where I can make a cut, insert the DNA fragment, and then close it. So now the foreign DNA would be part of the vector, would be part of the DNA. Okay? And creating a recombinant DNA. And by the way, the whole thing is known as recombinant DNA technology. That is manipulating DNA. Okay. Now, another feature that is important for plasmids to have is that they have to be selected, or they can be selected. What do we mean by that? It means that if a bacterial cell has a plasmid, I must select it versus bacterial cells that do not have plasmids. Okay, so this is known as a selectable marker, and usually it is what is known as an antibiotic resistance gene, okay, or a drug resistance gene. So if a bacterial cell has a plasmid, they must have this feature of being resistant to the antibiotic, so that they do not die, they do not get killed by the antibiotic. Okay, here's the thing, that's how we create a recombinant DNA. The idea here is that I take a piece of DNA from human cell. Okay, I make a cut of this DNA by echo R1, the restriction endonuclease that recognizes GAATTC sequence, and it makes a cut between G and A. Okay, now I take a plasmid from bacteria and I cut it with the same exact restriction endonuclease, echo R1. Remember here, since we're talking about the same endonuclease, the ends, the cohesive ends would be exactly the same. And they are complementary to each other, meaning that if I combine this DNA fragment with the plasmid DNA, eventually what would happen is that the ends would bind, would form hydrogen bonds to each other, right? Because they are complementary to each other. So you can have this plasmid DNA containing the DNA fragment of interest, the human DNA. Now, then I add a DNA ligase, and the ligase makes a phosphodiester bond, covalent bond, making or creating a stable plasmid vector or recombinant DNA. I take this plasmid and I put it back into the bacterial cells. Okay, so the bacterial cell would have its own chromosome as well as the recombinant DNA, the plasmid with the human gene. Okay, when I do this, now and I add an antibiotic to the media as well. That is, when I grow the bacterial cells, I add an antibiotic. Why? In order to select for the bacterial cells that have a plasmid. So bacterial cells that do not have a plasmid would die because of the antibiotic. They cannot replicate anymore. They cannot grow anymore. But bacterial cells that have the plasmid, they are resistant. They have the gene that makes them resistant to the antibiotic. Eventually, what would happen is that these cells would grow. They not only would amplify their own chromosome, of course, because they grow, they can also make multiple copies of the same plasmid. So bacterial cells would have at least one plasmid, and they can have multiple plasmids as well. So you can imagine that if I start with one bacterial cell having one plasmid, eventually after 24 hours, let's say, I'm going to have millions of bacterial cells because they grow really fast. And each one of them, if we assume that each one of them would have 5 plasmids, we're talking about 5 million copies of the same exact plasmid. And that's how we clone DNA.